Today, we're going to focus on distinguishing among different types of genetic patterns that can arise. So looking at things like incomplete dominance and codominance, and a little bit of some environmental factors. So stuff I think is kind of fun when we start looking at genetics. So there's a whole bunch of stuff here. I don't think I'm going to go through all of it today. We just don't have a lot of time. I know, cancel the gene linkage. And some of these are just a quick, short little blurb. We will look at some practice problems or examples, more or less, for the first three. And then the rest of this are just kind of some interesting patterns that we see, like here with the Siamese cat. The coloration of these little kittens is due to an environmental influence, and so we'll take a look at that. All right, so our first term here is called incomplete dominance. All right, and so when we're talking about incomplete dominance, we're looking at a true blending of the traits. So I know, and I talked with Wesley this morning about this because it's a confusing topic, I think. This is truly where when you have one phenotype that expresses a true thing like this red flower and another phenotype that's white, right? And those are given by the homozygous genotypes. If we mate those flowers and we end up with the heterozygous genotype, we don't see the red flower, we see pink. So somehow in how these traits are working together, they're actually blending and giving us an in-between version of whatever that phenotype was. So snapdragons, which are a type of flower, which I believe is what these are supposed to be, have this characteristic. So they're incomplete dominance. So incomplete means, if you want to put in your notes, that we're really blending the phenotypes of the homozygous genotypes. Another example, and if we put this into a Punnett square, is looking at Palomino horses. And so the Palomino color arises from this incomplete dominance. So if you were to mate a chestnut colored horse, that is the dominant homozygous, I guess, coloration there, with a cremello horse, which is the other version, and that's homozygous, we end up with this in-between color. Now, something I'm going to try to draw here on the slide just to point something out. You'll notice that the nomenclature that they use for incomplete dominance doesn't follow the rules that we established on Wednesday, right? So I'm not using big C for chestnut, little c for cremello. These are both just C's. And what we do is we write a number with it. So this is option 1, homozygous, and oops, forgot the 2. And this would be option 2, right? So version 2 of that same allele. So I find as I scoured some of the different biology texts, that when we talk about incomplete dominance, they usually use the capital letter C to represent that, and then use subscript numbers 1 and 2 to represent, I guess, the phenotype that's going to be presented with that homozygous genotype. All right, so that's kind of cool. So we've got this true blending. Any questions on that one? So again, this is, it's kind of like if you take the white crayon and color it over a different color, we kind of get that. Another example is with hair. So you have straight and super curly. And if you were to put that, mate those characteristics, you end up with wavy hair. So that's a human characteristic that follows this particular pattern. All right, so these are exceptions to Mendel's rules. Another one that we're going to look at here now is referred to as incomplete dominance with some traits. And this falls under something known as multiple allelism. So here we're looking at where you might have a larger selection of alleles for a particular trait rather than just two, right? So human blood type is one example where we can look at this. So humans have three common alleles that help determine our blood type. And there's the A, B, and O, right? Hopefully everybody's heard of those three types. And they're represented here by the letter I for incomplete. And so now we're going to use a superscript to kind of represent the particular trait. So this would be like the A blood type, the B blood type, and then we have this lowercase I that's representing O. And the reason for that is you can think of O as being recessive in a sense, but really if we look at it, and I'll show you here in a minute, what the A and B are representing are really proteins expressed on the red blood cells. And so if you have O blood, you don't have any proteins expressed. You don't have the A or the B. And so that's why it's represented by this lowercase I. So oops, I went the wrong way. So if we look at this picture here, we can see that they're showing us the cell membrane, right? So if you remember, this is like our phospholipid membrane here. And on the surface, we have these polysaccharides. So I'm sorry, they were sugars. And you can see that the top sugar is being represented here by red, blue, and then this one is completely missing. And so this is representing these different alleles for the blood type. And so if you have the genotype, so you're still going to inherit two alleles, right? One from each parent. So we will be able to still write a genotype for blood type. We're going to see that if you are A blood, right, that you may have the genotype, right? Capital I, A, capital I, A, right, is going to be an option for that. But you can also see down here, right, I have another A blood type. Whoops, I don't know what my pencil's doing. Okay, so we have another A, but notice we have IA and then lowercase I. So here, this individual inherited an A allele from one parent and an O from the other, but their blood type is going to be expressed as A. So like when you go to donate blood, right, and they need to know your blood type, you could have either of those genotypes and it's just going to show up as A blood, right? That's not going to tell you if you're AA or AO. It's just A blood. The same is true with B. So if we look over here, we see that B blood can also be expressed by a homozygous pairing of both parents donating the B allele, or it could be expressed by a heterozygous combination. So B blood and O blood, and this would basically, you would have B blood. Now, if your parents were A and B, and one donated the A and one donated the B, you would end up having AB blood, meaning that on top of your red blood cells, you would actually have both of these polysaccharides sticking out. So you have both of them. But if you have O blood, that means that you inherited the little I from both parents. All right. And so your cell isn't going to be expressing that top polysaccharide at all, right? So it's just not there. All right. Let's see, there's a good question in the chat. Would phenotypes in a population be skewed towards being blended over time, or would it stay random? That is an awesome question, Riley. And I actually have a slide coming up here that kind of shows us the percentage of the different blood types. And it's kind of interesting how it's come about. So I'm going to save my answer for that till we get to that slide. So So here's just another example of, how we could represent in a picture what our blood types look like. So the A and B allele really produce, what they're producing is an enzyme that helps to make that particular polysaccharide on that glycoprotein. And so if you have A and B glycoproteins, you kind of look like this blood cell down here on the right. So you've got both of those types of proteins being expressed, where in O blood, again, you didn't inherit the allele for that. So you don't have any of those glycoproteins being expressed on your blood cells. Oops, sorry, I forgot to copy the password down. All right, so why is this even important? Why do we care about what blood type people are, right? We care because our bodies care. If you were like A blood and needed a blood transfusion and somebody gave you B blood, that's not going to go very well for you. So why is that? Why is it so picky? Well, the thing is that our body makes these little things called antibodies, which I'm sure people are pretty familiar with these days, right? This whole COVID thing and getting vaccines, right? The whole purpose of the vaccine is to help your body build antibodies against that virus. Well, our bodies also make antibodies against these glycoproteins that are on our blood cells, all right? And they're making them against the glycoproteins that you don't have, right? So I have O blood. So my body, it would make antibodies against both A and B glycoproteins. It's not going to like either of them, right? And so if you have A blood, your body will make antibodies against the B glycoproteins. Whereas if you have type B blood, your body will make antibodies against the A glycoproteins. Makes sense to everybody? So it's that they're going to make the antibodies against the thing you don't have. And so if you have AB blood, that's actually really good because your body's not going to make antibodies against A or B because you have them. And so you don't want to attack your own blood cells, right? So that would not be good. So people with AB blood then are able to accept blood from any donor. And we're just going to keep it with this. There is also the Rh factor, the plus, for A positive, B negative type of thing. But if we just focus on the AB at this point, they're happy with any type. If you're O blood, you can donate it to everybody, but you can only accept the O blood, right? So, because you're making antibodies against everything else. All right, so here, this slide does show both the antibody, the antigen for A and B, as well as what's known as the Rh factor, which is what gives you your positive or negative, right? And it's Rh for the rhesus monkey, where it was discovered. This is actually a more important protein, especially for women when they're pregnant. It's very important that they know if they're positive or negative, because the body will get very violent, I guess, against if you are have negative blood type and your fetus is positive, it will start attacking, right? It will start making antibodies and attacking the blood cells and stuff in you, and that's not a good thing. So this image is just kind of showing us if you're AB positive, so you have all the goodies, right? Your cell surface is pretty busy there with A and B antigens. So an antigen is the thing that you're expressing. And then it's also got the Rh factor. So we can see all of these pluses up here have the Rh factor added. And at the bottom, you can see that they don't have them. And so the antibody, you can see, is going to basically attack then the thing that you don't have. All right, so Haley, this is your, hopefully helps to start answering your question that you asked there about the percentage of, where is a population going to migrate. And so you can see here that in the United States, that roughly 42% of the population has AB blood, and 44% has, has A blood, and 44% has O. But the smallest percentage is actually with AB. So if you are AB blood type, you're actually quite rare. So it's kind of interesting how this happens, but it has to do, I think, with what the starting population has, right? So if most of the population, right, so almost, we're approaching 50% here, has O blood, and if those people mate with the people that have AB blood, you're going to end up, with offspring that have A or B. But again, our population is very heavy A-centered. And so it would take a long time to have this kind of increase so that the population would have that AB blood. And the reason for that is because this isn't like a dominant recessive thing, right? Where they're both being expressed. And so, nothing really gets masked too much in this. So it would take a while really to change these types of percentages, in our population, just due to the fact that, AB isn't very popular, right? If you look at the whole population. Now, this does change depending on geographical region, especially if you look globally. And I don't have that data. I should have pulled that for different countries. So they may have their percentage breakdown of blood types can be different. And the reason for that is because I want to say, trying to think, that the O type blood, it might have been a mutation, I think is why that is there. But there are lots of studies out there, and I don't know if all of them are, genuine scientific studies, but kind of show how diet and how, like, what was I going to say? That you have some type of immunity to different diseases and things, depending on the type of blood you have. Like some people say if you have O blood, like, you're more likely to get eaten by mosquitoes. I don't know. I have O blood and they love me, but I don't know if that's a true fact or not. But if it were, right, and you think about that, if I were to live in an area where malaria was a problem, right, and if I had O blood, right, and mosquitoes did like O blood, then I'm probably going to get sick, right? So I'm not sure that, but like I said, there's lots of studies out there that where they kind of look at, where did these different types of blood types really come from? And these are the most common. They're not the only ones, which is another interesting kind of fact out there. But these are the ones that you're going to see, most often. So just kind of interesting things there. We could spend like a whole class period, I think, talking about blood. It's really fun. So yeah, we got the most common type. Livia, for right now, I'm going to have to say that the O blood here in the United States is the most common blood type that we see. This is actually a funny story. So when I was, I think, in high school, my dad and I were joking around, and I was O blood, and he's like, well, you can't possibly be our child because I'm B and your mother's A. And he's like, so, you must be, switched at birth or something. And I was like, oh, no, let me show you using a Punnett square. I can show you how I could still be your child, right? So, you can use this on your parents, right? So I said, well, you don't know if you're double A. And I said, so I'm going to assume you're AO and mom is BO. And look, I can exist. So not quite a paternity test, right? But could put that out. And so it was kind of a joke, but and it was funny because I was able to find this Punnett square online, so I didn't have to draw it. But yeah, so it's kind of fun to play around with these. So it's interesting because if you know your blood type, and you know your parents, like even if it's just A or B, right, you can kind of go back, and if you know your siblings, and you can kind of try and figure out, you know, if you're AO or AB. So for us geeky science nerdy people, you know, this is something fun to do at, like, you know, family gatherings, right? Hey, let's do our Punnett square blood types, right? Show them what you've learned this semester. So you can have fun with that. The next thing I want to touch on real quick here too is, and this is your extra credit, right? For lab, you can do the sex-linked fly crosses. I wanted to just kind of go over the nomenclature for this because it gets a little bit messier than what we've been doing with the little pea plants. So here we typically do work with dominant and recessive traits. I'm not looking necessarily at like the codominance that we saw with the horses, or the incomplete dominance that we saw with blood type. So we're back to looking at, you know, dominant and recessive traits, but now we're looking at X and Y chromosomes. And the Y chromosome, as we know, is a lot smaller, right, than the X chromosome. So there's going to be information located on that X chromosome that isn't going to be represented on the Y, right? It's missing that piece. And so if we look at, and I believe this is obviously looking at flies because we don't have white eyes in people. If we look at the white-eyed male, we can see, right, a male has XY chromosomes, and the allele for having white eyes is recessive, but it's found on the X chromosome. All right, there isn't a counterpart for it on the Y chromosome. So even though it's recessive, we're going to see that here in the male. If we look, oops, sorry about that. If we look at the female who is homozygous dominant for red eyes, we can see, you know, that we would put the capital R there. So this isn't the most exciting Punnett square because it's going to give us all offspring with red eyes. So this recessive allele kind of is going to be hidden in these offspring. What's more interesting is if you have a female who's heterozygous, right? Because then you're going to see, like, so if I just pretend that this is a little r, we'll get rid of that big R, we would see that this would be a little r. And this one here, let's see, what am I bringing over? Yeah, this one here would also be a little r. All right, and so here we would see that this male would be have white eyes, and in this rare instance, you could actually have a female show up with the white eyes because the male had white and the mom was a carrier, and so you could end up with a female having this trait. But because a lot of these recessive alleles are carried on the X chromosome and don't have a counterpart on the Y, we see them expressed more in the male than we do in females. So in humans, this is stuff like red-green color blindness, right? So you can be a female and have red-green color blindness. You would have, you know, this genetic makeup here. But for a male, you've definitely inherited that from your mom, right? So she was a carrier for that. And so we see more of that in the male population. So these are also really fun to keep, you know, to do some of these problems with. And I think I've given you some in your TRAT today, so won't take up too much more time here. I want to try and wrap this up here in the next couple minutes. The next example is something known as pleiotropy. So this is a really funny term, but this is where a single gene has an impact on several phenotypes. So this is something we haven't looked at all. So this is getting into complex genetics here, but it's kind of fun. So these little guys here are nude mice. So these are the mice I used in my research as a graduate student. And the allele that had been manipulated was one that dealt with their immune system, so that they wouldn't reject cells from other organisms. So you could put human cells into a mouse and they wouldn't reject it. But when that mutation was made to allow that to happen, it also affected their ability to grow hair. So they're naked. It had nothing to do, directly with the immune system, but now we have these poor little nude mice that are a result of that manipulation. Another example of something that's an exception to Mendel's rule is something known as polygenic inheritance. So again, poly meaning more than one, so more than one gene in this instance. And so here is where multiple genes are affecting a single trait. So kind of the reverse of pleiotropy that we just saw. And so in humans, your height, your skin color, your body build are all examples of something that's polygenic. So here you have multiple genes that are giving rise to that one phenotype, right? So these get really, really complex as we look at those. So if you go on to take genetics as your sophomore level course, you'll get to explore a lot of these things a lot more. So just kind of giving you a little teaser here for some of the fun stuff other than pea plants, right? And then here's a really cool one. So the environment can impact gene expression. So we talked about, how you turn genes on and off in this unit or our prior unit. And so we can see that the environment can have a profound effect on phenotype. So I showed you the picture of the Siamese cats at the beginning of class. And that has to do as well with the environment. So the Siamese cat actually has a genotype to have dark fur all over its body. So it could be a black cat, right? However, the enzyme that is used to make that dark pigment is inactive at temperatures above 93 degrees Fahrenheit, right? So when it's really warm, we're going to see that pigment can't be made. And so if we look at our little Siamese cats here, the little kittens that were born are all white because they were just born and inside like their mom, right? The temperature is really warm. And so it's going to deactivate that particular enzyme. And so they're going to be born all white. If you look at the Siamese cat itself, it kind of varies, right? In its coloration, it's kind of like a light brown. You can see near its like chest region in the top of its head, where a lot of our blood flows, right, near to our heart and to our brain. That's where your body temperature is going to be a little bit warmer, right? And so the cat's almost white. It's not able to produce that black pigment in those areas. Whereas you go out to the extremities, like the ears and the nose and the tail, the feet, those are going to be able to express the darker pigment because the temperature is going to be lower there, and so that they can express it. So it's kind of cool how like the color of this cat is reflected by what its internal body temperature is. It'd be kind of cool if we had a gene like that right now, right? So everywhere you go, they didn't have to like zap you with a thermometer to see what your temperature was. Like you could have turned, some funky purple color and like, hey, yep, your temperature's good, right? So these cats are pretty awesome with how they do that. And you can see many, many more examples of that. I'm going to stop here. If you want to read about gene linkage, you can, but I'm not going to test you on that. And it gets a little bit more complicated. And I want to give you time to work on your genetics problems. So does anyone have any questions before we go into our breakout rooms today? I know I went through that really fast, but it's recorded, so you can always go back and listen to it, right? Any questions? All right, I'm going to go ahead, open up those breakout rooms and let you all work on those problems. If you have any questions, let me know and we can hop in and help you out. Let me get some of these folks put to the right rooms, I hope. That is weird. Okay. Where goes to 13, 12. Did that go? All right. And Bailey, you are in 7, is that right? Okay, and Amelia, there it goes. It wasn't giving me the option for team 1. All right.
Yes. Miss Arrington.
Yeah.
I can be the scribe for the week. I've actually been driving home all morning.
Oh.
I just can't do it like now because I don't have my laptop, but I can.
That's okay. Wesley also signed up, so your team actually has 2.
Okay.
So yeah, you're good to go. Are you able to still join your room to help them out?
Yes, ma'am. I can do that.
All right, awesome. Thanks for letting me know, Ike.
Yes, ma'am. Of course.
Hey, Ben.
I got put in the wrong breakout room.
Oh no, that's not cool. Which room did you get put in?
I have no idea, but I'm on team 2.
You're on team 2. Okay, let me find where you ended up. So I have to. Where did it put you? There.
Oh, it says team 12.
Yep. Let's see. Did you get a new invite?
Thanks.
Yep. And I had made him the scribe. Hartley, are you here with us? Okay. Finally got where I needed to go, or she got where she... Feels like a marathon today.
Oh, I know, yeah. No, I actually got my second vaccine this morning, so I was like out all morning. Because of course, like since I'm going out, I'm like, I'd rather go to the grocery store now so I don't have to leave my house again.
Yes, yes. You'll probably be very tired tomorrow from what I don't know, like the people I know have gotten their second ones. I don't know. I guess it depends if you've got Pfizer or the other Moderna or whatever it's called.
Yeah, this one's Moderna. But we'll see. I don't know. I don't know if anybody in my family who's gotten it yet has felt anything.
Okay.
When I got the first one, I felt really tired and felt a little nauseous at the end of the day. But I think really I was just annoyed and didn't want to do work anymore with my group.
You have an
like,
excuse,
okay.
right? Like
Honestly, it was great because the 2 people, or no, one of the people who hasn't been helping with anything was there. So I was like, okay, I feel sick. Can you turn this in and finish it up? So that was nice.
Cool. Yeah, I've been doing like tech support this morning, office hours.
Oh man.
Putting in extensions for students. It's like everything and everything under the sun. I'm like, okay.
Friday, huh?
Yeah. Yeah, I was super excited that my I guess I should stop